Dear all, I'm going to present the study on immunological properties and protective effect of DNA vaccine based on human papillomaviruses E6 and E7 protein. Human papillomaviruses are responsible for more than 95% of cervical cancer cases, and major risk factor is infection with HPV genotype 16. While HPV infections and associated cancers are preventable by prophylactic HPV vaccines, there is a need for therapeutic vaccines to treat chronic HPV infections and associated cervical cancers. And promising vaccine components are viral oncoproteins E6 and E7, which play an important role in the malignant transformation of the infected cells. And our aim was to evaluate immunogenicity in mice of DNA immunogens encoded consensus HPV 16, E6, and E7, and evaluate their potential to protect mice against E6 and 7 expressing tumors. Compile the following tasks. First, information-based design of DNA vaccine candidate. Second, synthesis and characterization of E6 and 7 DNA vaccine candidates. And last, experimental testing of the vaccine candidates in the marine models. To obtain the consensus sequence of 6 and 7 genes of high-risk HPV, our group performed an analysis of viral isolates from the cervical smears and cervical tumors obtained from the patient with cervical neoplasia and cancer. And HPV-6 was found to be the dominant genotype in both patients' cohorts. E7 oncoprotein was found highly conserved, while E6 contained several single amino acid substitution uniques for each of the samples. And nearly 50% of isolates contain substitution arginine to glycine in position 17 and leucine to valine in position 9. And designing the DNA immunogens, the one of the E6 substituting variants was chosen. Both E6 and E7 genes were inactivated by introducing to the point mutations and cloned into eukaryotic expression vector PIVAX. Immunization of the laboratory mice was performed by intradermal injection of the plasmid DNA solution followed by electroporation using prime boost strategy. And the experiment endpoint, mice were euthanized and the spleen sites and sera were collected to test the immune response. Groups of mice were immunized by E6 and E7 DNA immunogens alone or by combination of separate E6 and E7 DNA injection, using as a control immunization with plasmid, vector, PIVAX, just empty DNA. And the panel of the peptides derived from E6 and E7 proteins was used in the immunogenicity test, including the peptides containing the substitution discovered and aligned with the E6 genes viral isolates. T cells immune response was evaluated by stimulating the splenocytes of immunogenic mice with synthetic oligopeptides and analyzing by flow cytometry the populations of T helpers and CTLs responding by expression of cytokines interferon gamma, interleukin 2, and tumor necrosis factor alpha alone and in these combinations. The specific expression of interferon gamma cytokine was detected only in T helper cells of mice immunized by single E6 and stimulated by E6 peptides, and no response against E7 was detected. The specific secretion of interferon gamma and IL-2 cytokines in response to the peptide stimulation was detected only in T helper cells of mice immunized with E6 DNA. It was induced by stimulation with peptides derived from the N-terminus E6. The cells expressing triple cytokines after stimulation with E6 peptides were typed as CD4 plus T helper cells. But cytotoxic cells were not reactive. But among the peptides containing the amino acid substitutions, only one representing E6 sequence used for immunization was immunoreactive. indicated that substitution arginine to glycine in position 17 may lead to immune escape. And to evaluate the protective effect of HPV immunogenes, mice were immunized as in previous experiment and challenged by injection of tumorigenic cells expressing E6 and E7 genes of HPV and labeled by bioluminescent marker, firefly luciferase. And the follow-up of the tumor development was performed using in vivo imaging and morphometric measurements. The tumorigenic cells used for challenging are murine mammary adenocarcinoma 41 cells labeled by firefly luciferase and expressing HPV 16 E6 and E7 genes. The two clones of tumor cells were used. More aggressive EEH6 with high level of expression of E6 mRNA and less aggressive EEB2 with low level of expression of E6 mRNA. The development of the tumor from injected cells were assessed by in vivo imaging, registering the bioluminescence signal emitted by firefly luciferase after injection of the substrate luciferase. And here we compare, there is the same tumors at the day 7 and at the day 25 after injection. So this is a growth of this bioluminescence signal. According to the bioluminescence data, at the early stage of tumor development in vector immunized mice, more aggressive H6 cells expressing higher level of HPV, and the genes were growing faster than less aggressive B2, expressing low levels of HPV genes. But at later time points, the growth rate of both clones became equal. The reduction of the final base of the tumor caused by deimmunization was shown for the more aggressive H6 clone, expressing high level of HPV genes, but not for less aggressive B2 clone, expressing low level of HPV genes, as if the latter were not seen by immune system. And using the bioluminescence imaging, we could demonstrate that DNA immunization with E6 restricted the capacity of tumor cells to infiltrate the liver. And the reduction of the metastatic activity in the liver caused by E6 DNA immunization was confirmed by histological analysis of both H6 and B2 tumor cells expressing HPV genes. So we can conclude that only immunization with E6 DNA provides the specific cellular immune response. The epitopes of CD4 T cells are localized in the N-terminus and central region of the E6 protein. And no cross-reactivity was found between R17 and G17 variants of E6 proteins, indicating this the substitution may represent an immune escape mutation. And immunization with HPV DNA partially suppressed growth and motility of murine adenocarcinoma cell expressing HPV 6-7 genes in syngeneic mice and reduces the number of liver metastases. And obtained results are important for the design of therapeutic HPV vaccines. I am grateful to the all contributing in the study and supported it. Thank you for attention.